Hello, dear friends. Today we will start with another important system of our body, that is the visual system. And in this lecture, we will discuss about the electrical activity of the visual system. So let's begin our discussion with the description to eye. Eyes are organs of the visual system. They provide us with vision, the ability to receive and process visual detail. The eye and its major components are as shown in the figure. Light enters the front of the eye at the cornea. Behind the cornea exists a transparent fluid called the aqueous humor. Its main function is to make up for the absence of vasculature in the cornea and lens by providing nutrients and oxygen. The aqueous is also responsible for generating a pressure of 20 to 25 mmHg, which inflates the eye against the relatively inelastic boundaries provided by the sclera and choroid. Further, the lens is located behind the aqueous humor. Its shape and refractive index are controlled by the ciliary muscles. The lens completes the focusing of the light begin at the cornea on the retina. Between the lens and the retina is the vitreous chamber, which is filled with gel-like transparent material known as the vitreous humor. The center of the visual image is focused on the retina to the fovea, where visual accuracy is the highest. The retina contains photosensitive cells in several layers of neural cells. This combination generates action pulses relative to the visual image, which passes out of the eye to the brain on the optic nerve. Further, this figure basically now we will little bit discuss more about the retina. This figure shows major elements of the retinal cellular structure. Light enters from the top and passes through the neural structure to the photoreceptors, which are the rods and the cones. Just behind the rods and cones is the retinal pigment epithelium, that is RPE. Its major function is to supply the metabolic needs as well as other supportive functions of the photoreceptors. The rods basically respond to dim light, whereas the cones contribute to vision in bright light or in color. Next step, we will discuss about how the signals are generated. The initial step in the translation of light information from a spot of light into an electric signal propagating to the visual cortex takes place in the photoreceptors, that is process known as transduction. That consists of cis-trans isomerization of the carotenoid chromophore, which leads to a transient change in the membrane potential of the cell. The result consists of a graded response seen as a hyperpolarization of the photoreceptor and an electrotonic current linking to the outer and inner segments. A photoreceptor is capable of transducing the energy of a single photon, that is about 4 into 10 to the power minus 12 ergs, into a pulse reduction of axial current of about 1 picoampere, lasting about 1 second with an energy equivalent of 2 raised to into minus 10 raised to the power minus 7 ergs. So thus basically you can see a photoreceptor serves as a photomultiplier with an energy gain of something about 10 raised to the power 5 times. The combined volume conductor signal from all photoreceptors contributes what is known as the late receptor potential, commonly it is referred as LRP. Then photoreceptor synapse with the horizontal cell and bipolar cell in what is known as triad. The signal transmitted via the horizontal cell results in the inhibition of neighboring receptor cells and hence an enhancement in the contrast occurs. The bipolar cell responds electronically with either a hypolarization or depolarization. The bipolar cell synapse with ganglion cells. This synaptic connection, however, is modulated by the amacrine cells. Further, amacrine cells provide negative feedback and thus allow regulation of the sensitivity of transmission from the bipolar to the ganglion cell to suitable levels depending on the immediate past light levels. At the ganglion cell, the prior means slow graded signals are converted into an action pulse that can now be conveyed by nerve conduction to the brain. The magnitude of the slow potential is used by the ganglion cell to establish the firing rate, a process sometimes described as converting from amplitude modulation to pulse frequency modulation. The region of the retinal pigment epithelium and the posterior portion of the photoreceptors, mainly rods and cones, is called the outer nuclear layer. Further, the region of contact of the photoreceptors with the bipolar cells is known as the outer plexiform layer. The main function of this layer is to be the signal processing. Since there are 100 into 10 to the power 6 rods and 6 into 10 to the power 6 cones, but only 1 into 10 to the power 6 ganglion cells, a marked convergence must take place in the course of signal processing. The bipolar and American cells form the inner nuclear layers. The region of contact of the bipolar and amacrine cells with the ganglion cells is known as the inner plexiform layer, that is basically referred as IPL. The amacrine cells play a role similar to the horizontal cell in the OPL, except that the amacrine cells act in the temporal domain, whereas the horizontal cells affect the spatial domain. Now let's see what are the sources of electrical signals in the eye, and also we will see some signals associated with that. The eye is a seat of a steady electric potential field that is quite unrelated to light stimulation. In fact, this field may be detected with the eye in total darkness and or with the eyes closed. It can be described as a fixed dipole with positive pole at the cornea and negative pole at the retina. This potential was not affected by the presence or absence of light. It was thought of as a resting potential. Such coronioretinal potentials are well established and are in the range of 0.4 to 1 millivolt. Eye movements thus produce a moving, or we can say rotating, dipole source, and accordingly signals that are measure of the movement may be obtained. This potential difference and the rotation of the eye are the basis for a signal measured at a pair of periorbital surface electrodes, and that signal is known as electro-ochlogram. The chief application of this signal is in the measurement of eye movement. Let's discuss something more about this. This figure illustrates the measurement of horizontal eye movement by the placement of a pair of electrodes at the outside of the left and the right eye. With the eye at rest, the electrodes are effectively at the same potential and no voltage is recorded. Now, if the rotation of the eye is to the right, results in a difference of potential with the electrode in the direction of movement, that is, then it will become positive relative to the second electrode. And ideally, the difference in potential should be proportional to the sine of the angle. And if we are rotating to the left, the opposite effect should result. As you can see in this figure also, if we are rotating to the right, that is a kind of positive pulse, and if we are rotating towards left, then it is a little bit of negative pulse. Electro-ochlography has both the advantages as well as disadvantages. The advantages of this technique include recording with minimal interference with subject activities and minimal discomfort. Further, it is a method where recordings may be made in total darkness or with the eyes closed. If I talk about disadvantages, the corneoretinal potential is not fixed but has been found to vary diurnally and to be affected by light, fatigue, and other qualities. Consequently, there is a need for frequent calibration and recalibration. Additional difficulties arise owing to muscle artifacts and the basic non-linearity of the method. Now, let's see another type of source and another type of signal. The lens of the eye brings the illuminated external scene to a focus at the retina. The retina is the site of cells that are sensitive to the incident light energy. As with other peripheral nerve cells, they generate receptor potentials. Thus, the collective behavior of the entire retina is a bioelectric generator which sets up a field in the surrounding volume conductor. This potential field is normally measured between an electrode on the cornea, that is contact lens type, and a reference electrode on the forehead. The recorded signal is known as electroretinogram or ERG. It may be examined both for basic science studies and for clinical diagnostic purposes. ERG, if you see the various sources of ERG that can arise in various layers of the retina, let's describe. These sources are distributed and lie in a volume conductor which includes the eye, orbit, and to an extent the entire head. The recording electrodes are at the surface of this region. For the ERG, one can identify the progressively changing layer from which different portions of the waveform arise, initiated by a brief light flash stimulus to the photoreceptors. The earliest signal is generated by the initial changes in the photopigment molecules of the photoreceptors due to the action of light. Then it gives rise to a positive R1 deflection followed by a negative R2 deflection. Together they make up the early receptor potential, that is ERP. This is followed after 2 milliseconds by a late receptor potential LRP, which combined with the remainder of ERP forms the main constituent of the A wave, a corneonegative waveform. Both rods and cones contribute to the A wave. However, with appropriate stimuli, these may be separated. For example, a dim blue flash to the dark adapted eye results in a rod ERG, whereas a bright red flash to a light adapted eye results in a cone ERG. Now another kind of maxima which is corneopositive is the B wave. To explain its origin, we should know what we mean by Muller cells. These cells are glial cells and have no synaptic connection to the retinal cells. The transmembrane potential of Muller cells depends on its potassium nearness potential, which is influenced by changes in the extracellular potassium. The latter is increased by the release of potassium when the photoreceptors are stimulated. In addition, the ganglion cell action pulse is also associated with the potassium efflux. The consequence of these events is to bring about a Muller cell response, and this is the basic source of B wave. Muller cells can contribute to B wave both from cone or rod, then it will be cone B wave or rod B wave. Another is C wave, which is positive like the B wave but otherwise is considerably slower. It is generated by the retinal pigment epithelium, that is RPE, as a consequence of interaction with the rods. The oscillatory potentials are small in amplitude waves that appear in the light adapted B wave. So this was all in the working of signals of in eye. Basically two signals we have studied, electro-ochlogram and electro-retinogram, and we have seen what are the basic sources of these signals in eye and how that is being generated in the retina when it is influenced by the entrance of eye. So this was all in this lecture and in this topic also. Thank you so much. This is one of the reference we have taken for this lecture. So thank you so much.